I think we can go ahead and get started. People can still trickle into the room after we've already gotten underway. All right, so I'm going to just say a few words before we let our speaker take over. Thanks to everyone for coming to this UMass Boston Biology Departmental Seminar. Once again, brought to you via the internet and Zoom. So I'm really pleased, very excited, in fact, to introduce today's seminar speaker, who's Dr. Jacob Berv, who's going to be talking about his work on macroevolution, let's say, and phylogenies with birds. So I've known Jake actually first in a entirely electronic form for at least 5 years. I have 5.5 years. I was looking back, I looked back today through my emails to identify our first email correspondence, which I think was originated from the our special interest group on phylogenetics, in which Jake was a participant, has been a participant for many years. And this is sort of an internet forum for resolving, for doing different types of phylogenetic analyses and things like that, for learning about how to do different types of phylogenetic analysis and communicating that with peers. So we corresponded on that on a number of different occasions, I would say, over those years. And then I think it was much later, actually, when I finally met Jake in person. And I think this might have been, well, I know this was at an evolution meeting. I don't remember exactly when, but it was several years after our first communication for sure. And at that time, he was still a PhD student. Now he's a postdoctoral fellow, as I'll mention in a second. And he was in one of the award competitions. I'm not sure for which one, but I think it might have been the Ernst Mayr Award, which is a prestigious award in systematic biology. He gave an amazing talk, in my opinion. He didn't win, and instead someone else won it, and they gave a much worse talk about research that I thought was relative. I want to identify who this was, of course. I guess we're putting this online, but no, the other talk was also excellent, I should say, officially. But I thought Jake gave the best talk in the group, but it's sort of that, actually seeing him give that talk and so on, and having that opportunity to interact with him that inspired me to invite him to the seminar series this semester. So just a little bit about Jake's background. He did his undergrad at Yale, and then subsequently earned his PhD in ecology and evolutionary biology at Cornell. And since defending his PhD, he's been a postdoctoral fellow in, I believe it's in Dan Roboski's lab at the University of Michigan. He's been really incredibly successful in his research program already. He's published a number of really excellent papers in top journals of our field, including in Nature and Systematic Biology, in Current Biology, and Molecular Phylogenetics and Evolution, in the OC. And also he's been recognized. I already mentioned that he was a finalist for this, I believe it was the Ernst Mayr Award. He also won the Publishers Award for Excellence in Systematics Research for a paper that he published in 2018 in the journal Systematic Biology, which is one of the sort of top journals in this field. And in my interactions with Jake, I've sort of discovered that he's a really nice sort of collegial guy, and so I'm very happy to host him here. I wish it was in person, but that's not what we're doing right now, so it'll have to be over this forum. So I'd like to thank Jake for agreeing to give a seminar here at UMass Boston under these sort of odd circumstances this semester. And so without further ado, I'll turn the mic over to him and thank him once again for being here.
Am I live?
You are good to go.
Awesome. Wow. Well, thank you so much, Liam. That was a beautiful introduction. I don't know what to say after that. I hope I live up to your expectations, I guess. Yeah, it's a real honor to have been invited. Yeah, I was always really, I thought of, so when I first started interacting with Liam was like, is still up here in the sort of stratosphere of sort of the phylogenetics gods. And so it feels pretty cool to have been invited by Liam to participate in this. And especially on this day, thank you for your attention. I hope I can keep your attention while all the interesting things are happening in the world today. So I'm a postdoctoral fellow at the University of Michigan. I'm associated with Dan Roboski, but I'm more closely associated with Stephen Smith, actually, and Matt Friedman at the Museum of Paleontology. And I think about phylogenetics and macroevolution and birds, and but I'm expanding out into other sort of taxonomic groups recently, and but I think mostly sort of through this through the lens of phylogeny. And whenever I sort of think about what I do, I like to sort of put it in the context of this sort of initial kind of thought that Darwin had a long time ago. And I love that he writes, you know, he sort of has a little sketch here, and he writes, I think, and he sort of sketches out one of these early schematics of what the tree of life might look like, and sort of the branching process that might describe how things are related to each other. And I like it because it sort of contextualizes how far we've come. And and so today we now think of phylogeny in much more sophisticated terms, and we can do things like estimate the population level processes that give rise to phylogenetic structure, and in pretty sophisticated ways. So we can sort of model the evolutionary process as a population level process that is sort of occurring within a species tree, and we can even model sort of introgression, hybridization events, and we can infer those things using genomic data. So we've come a long way since Darwin, and phylogeny also sort of, for me, is like, it's sort of like this thing that sort of contextualizes my own place in the universe. I sort of think of phylogeny as like this grand unifying sort of framework through which we can sort of understand and study biology. And that it sort of gives us a map of the biodiversity universe in a very real way. And so my research has been sort of structured around this idea and this lens. And so I think about systematics, molecular systematics mostly. And within that category, I think about molecular evolution, phylogeny reconstruction, divergence dating. So these are sort of three big domains within the field of phylogenetic systematics. And then also neotropical biogeography, applying the tools that I like to think about in systematics to particular questions in the neotropics. And so I spent probably about half of my dissertation was about neotropical biogeography. And unfortunately, I'm not going to talk about that at all today, because the more sort of macroevolutionary aspects of my work, I think, are more broadly relevant to this audience. But if you're interested in this topic, I encourage you to check out a preprint that we put out recently on this topic that I've been working on for a long time. So in systematics, in molecular systematics, the way we learn things is often through trying to connect, trying to use models to understand evolutionary processes, right? So the type of data we usually collect is in the form of genetic sequence data, and we then input that data into some kind of model. We use that model to reconstruct evolutionary history. We use that hypothesis that was generated for us to learn something about the evolutionary process. And then we sort of go around this circle, iteratively refining our models and our data and the way we collect and model over and over and over again. And through this process, we sort of iteratively refine our understanding of evolution. And this is also sort of one of the things that kind of inspires me to keep going in this field, is because I feel like I always have more things to learn. There's always, by sort of flipping back and forth between empirical and theoretical, there's always sort of, I always feel like there's new exciting things to keep thinking about. And in terms of the kind of things we can study with phylogenetics, paleontologists for a long time have recognized and sort of split up the types of change we observe in the fossil record into sort of two categories that George Simpson broke down into these two categories called tempo and mode. So the tempo of evolution is basically the speed of evolution, and the rate of evolution. And the mode refers to the way in which evolution occurs, the process that may be underlying the variation in tempo that we can observe. And in the fossil record, this is often sort of recognized as the difference between anagenesis and cladogenesis, for example, where you have change along a lineage, or you have speciation along a lineage, where you have the origin of new clades, or change along a lineage where you don't see origination of new clades. And that can be sort of gradual or punctuational, and the same can be sort of seen with speciation as well in the fossil record. And the observation of these two types of change, or these two types of processes, has been sort of distilled into these thematic, very broad thematic questions in evolutionary biology. One is this idea of evolutionary contingency, where that basically says that, that basically tries to understand to what extent evolution depends on a sequence of random chance events like mutation or environmental changes, potentially mass extinctions, and evolutionary convergence. To what extent is evolution repeatable? These are sort of two very classic questions that a lot of researchers have sort of focused on. And so a good example of this is the long-term evolution experiment, the Lenski experiment, where they have multiple independent lines of microbial sort of growth, and they're trying to understand, to see what happens across long periods of time. And so the debate is ongoing, and scale seems to be important in this sort of distinction between these two sort of ideas. And when I was doing my PhD, I kind of got interested in this idea about how these things might interact, and whether or not contingency can drive convergent patterns in evolution. And I got interested. I'll circle back to this in a minute, but to sort of go back into the idea of contingency, here's a famous diagram called the Sapkowski curve that illustrates the diversity of marine genera through time, back to 500 million years. And there are a couple of really interesting things about this. One is, I mean, the obvious thing is that there are these big dips, and you might know where this is going. But I was curious about this, so I actually downloaded the data for a relatively recent study of this. I guess not that recent, 15 years. And plotted it just to sort of look at it myself. And so what I've done here is I'm plotting the percentage, the origination intensity, so how frequently you're having origins of new genera relative in white relative to extinction intensity, and these are stacked on top of each other. And so if you look at the basically the extremes of situations when extinction intensity dramatically exceeds origination intensity, we find that there are these 5 events where really cataclysmic sort of turnover occurs in the history of evolution on the planet. And I'm interested in birds, and so I got really interested in studying the end Cretaceous mass extinction, because you can't really understand the evolution of birds, and I would argue the evolution of most vertebrate without understanding what the effect of this event was on their evolution. So this was a pretty dramatic event. We now think that about 66 million years ago, a large asteroid or comet hit the Yucatan Peninsula. It was about 10 to 15 kilometers in diameter. And one of the sort of more dramatic things it comes from a study from a while ago, but this researcher modeled how the sort of ejecta plume would have spread around the globe within a matter of hours after the impact. And so this little video that I'm showing you is basically showing you sort of a paleo map with an impact site in the Yucatan, and then within 8, 9, or 10 hours, the sort of firestorm that ensued has encircled the globe within 12 hours. And sort of one of the effects of this was to leave a layer of extraterrestrial iridium globally to deposit all over the world in this layer that we call the KPG boundary now. And it was discovered by Lewis and Walter Alvarez in a really famous paper from 1980. So backing up a little bit, so this event, so this asteroid was huge. We can really only study it. It took a long time to find it, actually, because most of it's been eroded down, but we can actually see it now from space using maps of sort of gravitational anomalies in the Yucatan Peninsula. And you can actually, so that we actually can measure it directly now. And in 2016, a group of geologists commandeered a drilling platform and drilled down into one of the rings of this sort of into one of the rings of the crater, down to the layer of the KPG boundary, to sort of study the composition of the strata at that level. And they found something really interesting, which was that even though that area is very rich in gypsum, the particular layer that the core that they extracted was almost completely absent. The gypsum was almost completely absent, which basically indicates that the sulfur compounds in that strata were completely vaporized and ejected into the atmosphere. And so they were able to estimate that this event likely ejected about 325 gigatons of sulfur and 425 gigatons of CO2 into the atmosphere within a day. And when I learned about this initially, I wanted to know how that compared to like the human amount of CO2. And it turns out that this is like more than a third of the total amount of CO2 that human beings have ever emitted. And this is like on one day. And so it was a bad day. And so there have been studies since then that have tried to understand the effect that this event had on global climate. And so here's a cool animation that shows, that tries to predict what the climate would have looked like within the months after this event. So you're going to see a countdown in the top left, months before impact, and the temperature in Celsius on the right. There's the impact, dramatic cooling, colder, 18 months, going down to -20, in equatorial regions. And only after 50, 60, 70 months are things sort of coming back to sort of normal temperature ranges. Really almost 90 months really is based on these simulations. And so this would have been bad for anything living on land, and most of the things living in the water too. And I had a chance to work with some paleobotanists a couple of years ago to try to think about what impact this might have had on birds living in trees. And this is sort of work that was initiated by Daniel Field and Antoine Berkovici. Antoine is a paleobotanist who studies the KPG boundary. And he had discovered a really interesting pattern, which is that if you look at fossil pollen from the latest Cretaceous, the latest Maastrichtian, you see this really nice sort of diversity of different forms. But then immediately after the boundary, there's like a complete wipeout. You only see these basically fern spores. So the diversity is completely obliterated. And so this is, we can look at this more quantitatively. And so here's this sort of down here, high diversity Cretaceous fauna. And then right after the boundary, you see this signature, this pulse of disaster flora, which are mostly ferns and relatives. And this is pretty cool because this is sort of what you see even today when the landscape is sterilized, like after a volcanic eruption. The first things to come back are ferns. And it's remarkable that this signature, it turns out, can be detected basically globally, which suggests that there were that there must have been global wildfires of a scale that we can't imagine. And so Dan Field and I were talking to Antoine about this, I don't know, 5 years ago, and we realized that at the time of the latest Cretaceous, there were a lot of early bird-like experiments that were living in trees. And so if all of the trees were demolished by this, then perhaps there is a macroevolutionary signal of the loss of trees in the phylogeny of birds. And so we took those data and combined it with an ancestral state reconstruction approach and also direct fossil evidence. And we argued that there is we analyzed these data in a way that allowed us to argue that indeed there does seem to be a strong signature of ecological selectivity on birds that's sort of baked into the structure of the tree of birds. Recently I've actually been working on asking the same question in mammals, and it turns out that the same signature is sort of observable in mammals too. And so these questions and a number of others got me really interested in this idea of trying to understand what the effect the KPG mass extinction had on structuring modern biodiversity. And in the last 10 years, there have been a number of phylogenetic reconstructions of major vertebrate clades that have hypothesized deep and close association between the boundary itself and the radiation of those clades. So for example, here's a mammal example, and it's not totally clear, but a number of these clades seem to be sort of diversifying right along the boundary. It's much clearer in this frog example, this bird example from 2014, and a more recent fish study. And all these studies hypothesize this close association between diversification, extreme sort of acceleration of diversification, and the KPG boundary. So what is the role? So this sort of like drove me to ask this question: what is the role of the end Cretaceous extinction in structuring biodiversity? And did this event drive convergent patterns of diversification? So these studies would imply that yes, it does seem like this event has driven convergent accelerations in the rate of diversification across lots of different clades. But I think in order to sort of really get into this question, and as it pertains to birds, which is my focus, we have to talk about molecular clocks, which are a type of statistical model that integrate genetic data with fossils to do two things. One, they estimate the rate at which genomes accumulate mutations, and two, they use that estimate of rate to try to estimate an evolutionary timescale. And these models can be relaxed or strict, and that sort of determines how variable the rates can be within your model. And to sort of explain or get everybody up to the same baseline, the way that we do this in practice, we usually start with an estimate of phylogeny that's derived from DNA sequence data, and just from that alone, we have no information about rate. We have information about relative divergences, and usually this is in the form of, or it's usually in the units of substitutions per site. So it is sort of a rate in that sense, but it's a rate relative to sort of itself. And so these branch lengths tell us about the amount of molecular evolution that has occurred along each branch. And if we want to convert this into units of time, we have to take this and give it something about that's real in time. And so we usually use fossils to do that. And the way we do that is we usually we take fossils and we identify morphological synapomorphies that allow us to determine where they fall in the phylogeny. We attach them to those particular points in the phylogeny, and then we use that to calibrate a model of a molecular evolution using different kinds of priors. So in this case, we're using a log normal prior that defines the minimum age of the fossil. So this is the age that the age that the node has to be at least that old, and then you sort of determine based on a number of sort of choices, the shape of that distribution to reflect the uncertainty that you have about how old that fossil might be. And then you take these priors, you take the tree, you take the data, and you run it through another model, and then you can sort of estimate a time scale. And that's what all of these studies are doing. They're making a lot of assumptions about how rate evolves, about how the branching process changes, and they look pretty good. And it looks like, if we want to ask this question about diversification, maybe this constitutes the test of that hypothesis. But the reality is that if we dig into the actual output from some of these models, there's really much more uncertainty here than these sort of presentations indicate. And I guess I would argue that this uncertainty is a major obstacle in sort of allowing us to address this hypothesis about whether or not the KPG boundary actually sort of had a significant impact on diversification of these clades. So there's a ton of effort of to try to ask, how do we reduce the uncertainty? How do we reduce the uncertainty? Do we use which markers do we use? Which models do we use? And that's all great and worthwhile. But I also would suggest that perhaps if we can think about why we might be getting uncertain answers, we might be able to learn interesting things as well. And to zoom in on the bird situation, here's a tree of the birds that I helped put together in 2015. And in yellow, I've indicated points where we've placed fossils that we use to calibrate this phylogeny. And the red arrow down here is pointing to the root of the tree, which we were not able to calibrate directly with the fossil because there are no fossils that we know of that we can place at that point in the phylogeny. A key point to recognize here is that there are no fossils in this major group that we can identify as occurring before the KPG boundary. These are all, within this major group, they're only post-dating the boundary. There is one fossil that we were able to place in this group, but otherwise, until very, very recently, there were no other fossils that we could really clearly put into this phylogeny to use for calibration points. And that was sort of a key thing that I'll get back to in a second. But anyway, we ran this analysis and we estimated the age of birds is about, the origin of birds is maybe between 66.9 and 79 million years. And this is quite a bit younger than many previous studies. And so some of those other studies have estimated a huge range of date estimates for the origin of modern birds. So here's an example from 2014 that did not use a constraint on the age of the whole tree, and estimated the origin of birds to occur at about 160 million years ago, which, knowing that the oldest fossils for most of the modern bird groups occur more recently than 66 million years, suggests that there's essentially that argues for 100 million year gap in the fossil record. And so there have been a huge amount of effort to try to sort of figure out what's going on with this uncertainty. And one of the things that would help us reduce that uncertainty would be the discovery of more fossils. And so one thing that happened actually just a couple of months ago was the discovery of a really extraordinary fossil. It's potentially one of the most important fossil discoveries in the history of bird paleontology, actually. It was discovered as an early diverging lineage that attaches to this group down here. And it's really the first unambiguous representative of an example of modern birds that we know to have existed before the KPG boundary. And so the analyses that I'm going to talk about do not take that into account. But for reasons that I'll circle back to later, I'm not convinced that it will make a huge difference in the story that I'm going to tell you. So, okay, so there are two sort of interpretations of this uncertainty that we observe in the bird in the bird situation. One is that the fossil record is giving us the right answer, that birds are really quite young and originated near the KPG boundary or slightly before it, or that the fossil record that we have is wrong, and the age of birds is much, much older, and we just haven't found fossils that we can place, or that we found fossils but we just can't place in the right place because they lack synapomorphies that we can place in the tree. So this is a really big sort of conundrum. And so you can say find more fossils. That would be great. If somebody finds a fossil that helps solve this problem, that would be great. But because we don't have those fossils yet, we sort of started to explore alternative hypotheses, and to get at that, we wanted to sort of ask, well, if the first scenario is true, if the fossil record is true, then that means that there has to be some kind of rate acceleration towards the base of this phylogeny that explains why these age estimates are so much older than the fossil record implies. But if the second hypothesis is true, then there's a misleading gap in the fossil record, which is possible. So because we can't actually test 2 yet, we decided to test for 1. And in order to do that, we sort of extracted the data from this divergence time analysis that we extracted the parameter estimates from this divergence time analysis that we did. And one of the things to note from this on the right here, so these branches are color coded and proportional to the inferred rate of molecular evolution. And so at a glance, the thing to note is just that there's a lot of heterogeneity. But when you sort when you take all those rates out and sort them from slowest to fastest, we find that there's a really interesting signal of body size that's sort of encoded into these rates. You might think that that's sort of an expected result. Fast things tend to on the short fast or the slow fast sort of life history continuum sort of makes sense that small things would be evolving faster. But I thought it was quite remarkable that from this analysis of genetic data and fossil calibrations, we can extract this signal of life history evolution across 6.4 billion years of branch length. And within that range, we find about 10 to 20 fold spread in between the fastest and slowest rates. So we wanted to get more mechanistic about this, and we built a model using a piece of software called coevol that is a pretty sophisticated molecular clock model that takes into account a number of different things, but also allows you to ask to what degree does the rate of molecular evolution co-evolve with a suite of quantitative traits. And so in our case, we got interested in life history traits because we had this sort of prior indication that body size was something interesting. And so we looked at a suite of life history traits, but the bottom line is that when we ran a whole bunch of models, the most explanatory model included body mass and metabolic rate, and also accounted for variation in GC, which is important for later. But we were able to explain with this model about 45% of the variation in rates with adult body mass alone at the tips of this tree, and a lesser amount of variation based on metabolic rate. And these are partial correlations that control for the effect of each other. And so here I'll go back a second. So the thing to just take to note from this is that the direction of the correlation that we checked with body mass was negative. So as you get bigger, your rate of molecular evolution goes down. And when we initially used metabolic rate, it scaled with body mass, so it was the same direction. But when we did a partial correlation, that accounts for the covariation with body mass, the direction actually flipped, such that basically mass-specific metabolic rate scales positively with substitution rate. So let me explain what I mean by that. So the nearly neutral theory sort of predicts that the substitution rate will scale with generation length, and so we think that what's going on with body mass is that as smaller things tend to have shorter generation lengths, and this is sort of what you expect, that this is the generation length effect. But the metabolic theory of ecology has a little bit of a more interesting kind of prediction, which is that it turns out that smaller bodied birds actually have higher mass-specific metabolic rates. And so we hypothesize that maybe the thing that's causing these correlations is sort of essentially a trait, both the effect of sort of demographic of a demographic signal derived from generation length, and also an intrinsic biological signal derived from the relationship between metabolic rate and mutation rate. And so we interpreted these patterns as sort of two things acting at the same time to both simultaneously increase the rate of molecular evolution in smaller bodied birds. And I should also mention that sort of at a higher level, that it does seem like body mass is a very good proxy for life history in general in birds. And I won't go into too much detail about that. But so this basically eventually has led us to this thing in the fossil record called the Lilliput Effect, which is this observation that a number of major mass extinctions have been associated with size reductions in major clades. Here's some examples. Actually, in the original Walter Alvarez and Alvarez paper in 1980, one of the examples they cite is Foraminifera, and they document a dramatic size reduction in the samples they look at. And other studies more recently have documented this in bivalves and vertebrates. There was a paper in 2012 that documented size reduction and dramatic size evolution in multituberculars, which is an extinct clade of mammals, in association with the KPG boundary. And so we started asking ourselves, can we use this Lilliput effect to maybe explain some of the uncertainty that we are observing in the bird, in our understanding of how old modern birds are. So in order to even, I think, ask this hypothesis, we have to we have to sort of check off two boxes. One, do small birds evolve faster? And then is there evidence for a size reduction associated with the Cretaceous Paleogene mass extinction in birds? And so I'm going to say we can put a check mark on the first one. And in order to address the second one, we took a little bit of a different approach. We wanted to look directly at the fossil record, even though it's sparse. And we started thinking of the KPG boundary as a body size filter for birds and how we might address that hypothesis. So in order to do that, we looked at this suite of sort of stem lineages that are related, very closely related to modern birds. These are the closest relatives that went extinct at the KPG boundary. And we took a sample of skeletal elements from those taxa that persisted up to the latest Maastrichtian, up to the right to the boundary. And we used those specimens to estimate a distribution of body sizes. And in order to do that, we used allometric scaling equations derived from comparisons with living birds. And when we do that, we found that this distribution of early bird diversity that was not directly along the line leading to modern birds, but closest relative to it, had a pretty large average size. And so there are some caveats with the way we did this, but in general, we think we did a reasonably good job because the phylogenetic relationships among these things aren't very well understood, so we're sort of constrained in that respect. And so what we wanted to do is compare this distribution to the earliest ancestors of living birds. And in order to do that, we have to do some modeling. So we don't have any fossils, or until when we did the study, we didn't have any fossils that we could directly place before the KPG boundary within modern birds. And the approach we took was a sort of a simulation-based approach where we wanted to ask, what are the relative likelihoods of models that allow size evolution to occur relative to this assemblage that we think was closely related to modern birds right before the boundary, compared to models that prevent size evolution to occur. And when we wanted to take into account the sort of best estimate of phylogeny at the time, various models of evolution, calibrations derived from fossils, and this assemblage of taxa that we wanted to compare. And so the way let me just sort of set this up for you. So these different models that we wanted to compare, the first model was sort of a static model where we predict that no size change occurs along these internodes. So we want to compare this assemblage of extinct taxa that went extinct to the earliest 3 nodes in the bird phylogeny. So this idea is there any evidence or signal of size reduction relative to this fossil assemblage? And in the first sort of null model, I guess you can call it, we don't allow any size evolution to occur. In sort of a medium conservative model, we allow about a 40% reduction in size to occur along these internodes. And then in our third model, we sort of constrain the model to allow for dramatic size reduction along these internodes. And when we run these models using some Bayesian techniques, we find that the dramatic size reduction model is dramatically preferred over the other two. And so this is an interesting way of testing that hypothesis indirectly, and I think is consistent with the idea that a Lilliput effect may be acting at this point in the history of birds. And again, it's an indirect test because we don't have that many fossils we can use to directly compare what's going on with birds. And so this kind of approach may be the best we can do until our fossil record improves. So here's a closer look at what the actual model output looks like. So here's an example of an unconstrained analysis where we're comparing our, on the left here, this is this fossil assemblage that we want to compare to the earliest diverging nodes within birds with a median mass of about 1 kilogram. And then here are the 3 sort of deepest nodes in the bird phylogeny. And we find that there's evidence for, at least based on these models, evidence for about 70% size reduction relative to the mean of this assemblage. And so this is kind of cool and sort of consistent with the idea, but we we wanted to take this a little bit further. So if we accept that the fossil record sort of requires a younger age for birds, that also requires that there are higher rates in those early diverging lineages. And if that is true, we can take that information into account when we actually do our ancestral state reconstruction. And when we do that, we find that the degree of size reduction along those internodes becomes even more extreme. And so ancestral sizes that are conditioned on substitution rates directly imply a more extreme size reduction than a fossil calibrated analysis alone will do. Was there size reduction associated with the KPG boundary? I think maybe. I think that there are a number of ways to go about this, and we went with a sort of simulation-based approach that I think is interesting. It's not definitive, but I would also say that if you believe the fossil record is correct, then we can make a conditional statement, which is that if the fossil record is correct, the fossil record requires that there are high rates because of the young age. And if that's true, then I think we have to actually accept that there had to have been size reduction because of that correlation that we observe between molecular rate and size. So again, so I mentioned earlier, there was this incredible discovery a couple of months ago, and I've been thinking about how this might impact the major conclusions from that original study. And so the lead author on this is a guy named Dan Field, who is also my co-author on the work that I just told you about. And so he was thinking about this too. And so if we look at a distribution of body masses from within the group that this new fossil, from where we think this new fossil occurs, we find that there's a huge spread of masses within this clade. But if we put this new fossil onto this distribution, we find that it was small. And so this certainly isn't proof that our hypothesis is correct, but it is indeed consistent with the idea that that high rates would have characterized these early branches of the bird phylogeny. So that was pretty cool. So in summary of this section, Jeremy Bellew had a paper in 2015 looking at angiosperm phylogeny and asked some similar questions. And their conclusion was that these analyses do not prove that the younger age estimates based on the fossil record are correct, but they do suggest caution in accepting the older age estimates obtained using current relaxed clock methods. And I think that that is a fair statement to make about this study as well. So the Lilliput effect may have contributed to this analytical uncertainty we have in birds, and has also potentially contributed to to our inability to test this larger question about convergence and contingency. That was a big chunk of my PhD. We published that in 2018, and then I switched gears for a little while and I worked on neotropical biogeography. I've been working on that for a couple of years. But in my postdoc, I've sort of switched back to more of these macroevolutionary questions, and I got really interested in not just these questions of tempo, but questions of mode. And so I've been working with Stephen Smith, who is developing some new methods to sort of get at this question of whether or not the mode of molecular evolution may be evolving in the clade of interest that you're studying. So Stephen was inspired by this study that came out a year ago for the automatic generation of evolutionary hypotheses. And I'm not going to go into the details of how this works, but essentially it allows the model parameters and the model itself to evolve on your phylogeny and does a sort of simultaneous optimization of all these things. And Stephen was inspired by this to develop a new technique that would allow us to ask this question about molecular evolution, not just modes of morphological or character evolution. And so I wanted to use this new tool to ask whether or not the evolutionary mode of molecular evolution, how does the way DNA evolves, whether or not that is associated with the KPG boundary itself as well. So just to give full credit to this method, Stephen Smith, Nathaniel Walker-Hale, and Caroline Perrins-Fukuchi are the main developers of this tool. And essentially what it tries to do is take a standard substitution model, in this case a GTR model, which is characterized by 6 rate parameters that reflect transition rates among nucleotide states and 4 parameters that reflect equilibrium base frequencies. And it allows those parameters to evolve along your phylogeny in a discrete way. So the idea is that it will allow you to potentially identify mode shifts. So how, when, and where does the model itself change on your tree? And to explore this, I've been using a new data set that I've been putting together with Sonal Singhal. That's a reassembly of the original Promedal data set that was an anchored enrichment data set. But the endpoint of this has been that we have a new data set of about 700,000 base pairs with very low missing data, and it's annotated very nicely to introns, exons, and UTRs. And so here's some very, very preliminary results from using this new technique that Stephen and colleagues are developing. When we run this model on an analysis of this entire concatenated data set, we find that, that 8 regimes are detected on the bird phylogeny. And most of them detect a transition transversion ratio of about 5 to 1. And the thing that seems to be really changing among these regimes is the equilibrium base frequencies. So another thing we can get out of this is which are ancestral and derived, and then we can plot. I'll show you what this looks like on the tree in a second, but just to point out some interesting things that are coming from this. If this was our sort of ancestral nucleotide evolution sort of state, all of these 7 states are being derived from that state. So there's dramatic evolution in some cases from a state of low GC content to high GC content, for example. And these are switching as clades are originating. Because we're trying this on a bunch of different topologies and a bunch of different ways, I'm just going to show you one example of this that is certainly not the final example of this analysis. But the pretty cool thing that seems to be emerging from this is that there does seem to be a signal of major model shifts at the origins of clades that are hypothesized to have been associated with the Cretaceous-Paleogene boundary. So I think that's unfortunately all I will be able to say on this for today, because I just did this analysis yesterday and I wanted to share it with you. But it does seem consistent with this idea that the KPG boundary dramatically influenced life history evolution in birds. So if we go back to this question about contingency and convergence, I think we can sort of say a couple of things. At least on its face, it does seem like a number of vertebrate clades were dramatically affected by the KPG boundary, and in some way, but I think actually understanding how that occurred is going to require a much better understanding of the nuances that are occurring within each of these groups. And the studies that I've done have been sort of focused on whether or not the models we use are fundamentally adequate to address this hypothesis. And in the case of birds, I think the answer is not really, and it's mostly because we need models that can account for the sort of systematic components of rate variation that are driven by life history evolution. So, for example, if the rates in your phylogeny vary systematically with life history evolution, or with body mass evolution, for example, then that is going to cause systematic biases in your estimated branch lengths, which is going to cause systematic biases in any kind of downstream comparative analysis that you perform. And so I'll loop back to that in just a second. But anyway, nonetheless, I think that there is some evidence, there's some reasonable evidence that contingency can drive convergence at this scale. And I've shown you some evidence that this is the case in birds and mammals. It's probably true in frogs and other groups as well. We have reasonable evidence of morphological convergence within and across lineages that survived the KPG boundary. There's direct evidence in mammals, in multituberculates, direct evidence in Foraminifera, indirect evidence in birds from the studies that I've done. And there's additional evidence from other groups as well. So one of the things that I'm getting more into as a postdoc is the question of whether or not contingency can drive convergence at a molecular level. And the studies that I've shown you today are sort of, and we're not quite there yet, I think, but we will be able to address that question soon. And this brings me to sort of maybe a higher level statement question that I think maybe we should think about, which is that we spend a huge amount of time and effort trying to estimate how old clades are using molecular clock models. And there's an enormous amount of uncertainty that's derived from the modeling process itself, but there are also these systematic sources of error that we haven't accounted for yet. And so while I do think that we can learn really, really interesting things by trying to start to account for those sources of systematic error and actually test hypotheses that we could not test before, maybe we should start asking a different question, which is that what must we believe about how molecular evolution occurs in order to either believe or not believe the fossil record? So for example, if we believe that the fossil record, that the signal from the fossil record in birds is telling us the truth, then that requires high rates of molecular evolution near the base of the bird phylogeny. And then that also requires potentially dramatic life history and size evolution associated with those clades as well. So there are a number of really interesting things that I think can be derived from this. So I think that what this is going to allow us to do eventually is this better integration of physiological ecology, micro and macroevolution is going to allow us to understand, number one, understand the qualities of extinct lineages better. And in some cases, for lineages for which we may not have any fossil record, I think that this is eventually going to help us estimate evolutionary timescales more accurately and with less bias. And through that, we will basically improve our understanding of diversification, because the current methods that we use to study diversification do not account for this source of systematic error. And that's something that I'm exploring as a postdoc as well. So yeah, and then this is going to help us understand phylogenomic conflict. Why do different estimates of phylogeny give us different age estimates, for example, in birds? Could it be because of life history evolution in certain, and whether or not that's occurring more in certain markers than others? And I think that there's also some very interesting, exciting approaches being developed to use these same ideas to identify candidate loci that are associated with complex quantitative phenotypes. So that's all I have for you today. I'd like to thank all the postdoc advisors that I'm associated with: Stephen Smith, Matt Friedman, Dan Rubosky, and Ben Winger. They've been really great supporters during this difficult time. My PhD committee, Irby Lovett, Amy McCune, and Anurag Agrawal, and then also the advisors that I worked with as an undergrad, who are still, all of these people are still close collaborators today. And with that, I'd be happy to take any questions.
Thank you, Jake. That was an amazing talk, super interesting. I made tons of notes and I will have a couple of questions, I'm sure, but
sure.
I think I'd like to allow others to ask questions. So if there's a Q&A box at the bottom of your screen, you can put a question in there, or this actually, we might try, if I can see if I can turn on the mic of anyone who wants to ask a question. I'll try and do that if that's possible. So you can also just indicate that you want to ask a question, and then I'll turn on your mic if I can.
Yeah. So Ron Eder asks, thanks, is there a difference in GC content of birds of different size? Stay tuned. Yeah, so it's a good question. It's not totally clear yet. It does seem like there is, the short answer is probably, but it depends on the type of genomic element or the type of marker that you're looking at. So it seems like this is more of, it seems like that signal is stronger in exons than it is in introns, for example. And I don't really know why that is yet. I have some ideas, but the short answer is probably, but it's complex.
Okay, there's another question here. Do you want me to read it?
No, I got it. It's okay. Quick question: are the distributions used in the molecular clocks independently chosen? Yeah, that's a good question. So the yeah, exactly. So there is when you're doing these kinds of divergence time analyses, there's a lot of sort of art to it, and it really depends on the expertise of the paleontologist that you're working with, I think. And in my case, I'm not really a paleontologist, and so I rely on the expert opinion of the paleontologists who help me choose the fossils to use for calibration. And so the shape of those distributions we can choose the shape of them independently for each calibration, and the shape is usually derived from an expert opinion on the likelihood of where a fossil, where the actual origin of a lineage would have occurred. So it's sort of a nuanced question, because it sort of depends on whether or not you only have one fossil or a series of fossils for a particular lineage, or if you have, there are some quantitative ways of estimating the shape of the distribution that you should use. That in and of itself is a whole big problem and a big question to deal with in the field of understanding how old clades are.
Okay, the next question, I'll read it off. It's from Rob Stevenson. He says, do we have fossil DNA sequences for birds? Will DNA sequences from fossils eventually help reduce our uncertainty about the transitions that occur?
So it's a great question. So the DNA has a half-life of about 500 years. So the oldest DNA that we'll ever be able to get, even under perfect preservation conditions, is maybe a couple of million years. And that'll be very fragmented. So the kinds of timescales that we're talking about here are way, way older than any DNA molecules are likely to be preserved. There's a little bit of a caveat there, which is that it does seem like protein amino acid sequences may actually be able to survive across vast geological time. And so it may be possible one day to get enough of that kind of information to make some inferences about what was going on, but not right now. And the thing that I'm actually more that I think is potentially more interesting in the short term would be to actually use estimates of body size from fossils to calibrate the rate of molecular evolution, not just the age of a particular group, but to actually calibrate the rate at which evolution is occurring along a particular branch deep in your phylogeny. And that should actually, I think that would actually help us reduce the uncertainty in some areas. But of course, then that requires understanding the relationship between size and rate, but that's not always very clear. And a lot of it doesn't, the models we have so far don't explain all of the variance. And so there are issues there, but I think that is, there's a potential way to improve things there as well.
So I'll ask one question. We're going to transition over to the happy hour social, and then everyone else will have also additional chances to ask Jake more questions. I'm just going to ask one question, which had to do with sort of, I think you presented a really strong case that those, I mean, I think it almost goes without saying that a sort of date of 160 million years and no bird fossils for 100 million years is sort of pretty implausible. And sort of you presented a pretty persuasive case that that's unrealistic. But then you also tried to link it to sort of a mechanistic basis for a shift in the rate of, or changes in the rate that you associated with sort of metabolism and body size. And you also talked about the nearly neutral theory. And so I was just wondering, like, so the nearly neutral theory has to do with effective population size.
Right.
And so plausibly in a mass extinction event, you'd have sort of really dramatic shifts in effective population size for lots of lineages simultaneously. And so I wondered if that's sort of where that.
Yeah, so I think that's playing a role too. I'm actually kind of confused about this pattern, because you would predict that that smaller bodied birds would have larger effective population sizes, and yet we still detect this very strong signal of increased rates in smaller bodied birds relative to large birds. And so that seems almost like a paradox that I don't really know what the answer is to. But nonetheless, we do seem, the signal I think is very real. It's a very strong signal of smaller things evolve faster, regardless of population size. And so it's more complex than one. I think one body of sort of one theory is not going to explain it all. I should also say that the type of DNA sequence that we used in the study that I talked about was mostly exon-rich data, and so there is. I think that it'll be interesting to do some follow-up studies on different types of genetic sequence data, using this new data set, for example, that we're developing, to see if the same kind of patterns are evident on introns and UTRs. I would expect that those patterns are going to be less pronounced in non-exons, and how that relates to nearly neutral theory is also going to be really interesting with respect to mutations that we think are neutral or not. But I think that the way we the way we sort of interpreted the signal was just certainly with a sort of a post hoc interpretation, and we tried to think of how can we explain this, and I think that the idea that there are these that there are processes that are being driven by demographics and by population size and generation length is plausible, as well as this idea that there are intrinsic biological processes being driven by changes in metabolism as well, and then they might be acting on different things, right? Because the substitution rate is sort of the fixation rate, right? Which is what we expect to be related to population genetic processes, but the metabolic rate hypothesis has to do more with mutation rate, which is the rate at which mutations are generated, and those rates are not necessarily the same, and so there's a lot of complexity there, and it's hard to parse out. It's hard to parse out causality. There is an approach using. I have seen one paper that used phylogenetic path analysis to ask a similar question in frogs, but I haven't been able to apply that yet.
Okay, that was excellent. So thanks again, Jake. And basically now we're going to disband this webinar, but I invite everybody to please consider attending the happy hour, where we're going to have a more sort of casual conversation where we can sort of talk more about this talk as well as about other things. And the attendees of the seminar will also have a chance to meet Jake and get to know Jake a little bit. It's BYOB, unfortunately. And the link has been posted. I believe it's in the chat. Is that visible to everyone, Brooke?
It should be, yes.
Okay.
The other thing I wanted to add is you can feel free to take a 5-minute break, Jacob, or whatever break you need to take.
I'll go grab I'll grab a beer actually as well. I'll come right back.
Yeah, it's a different link though for everybody to know. Yeah, so you have to we'll close this, and then you'll have to go to the link that we sent you before, Jake, and that everyone else should be able to see in the